A warm welcome to everyone who has dialed in. Thank you for taking time off your busy schedule today to join us in this webinar. My name is Yuan Lu, and I'm from DXD Hub, and I'll be your host today. We have lined up an exciting program to update you on the latest developments on COVID-19 vaccines, immunity, and diagnostics. This event is jointly organized by the Diagnostics Development Hub, or DXD Hub for short, and the Foundation for Innovative New Diagnostics, which is also known as FINDX. To our audiences at the webinar, you'll be glad to know that we have had an overwhelming response today. We have more than 700 people signed up, and more excitingly, one-third are colleagues and friends from the ASEAN countries. Let me give a brief introduction on the program before we start. Firstly, we will have Dr. Sydney Yee to provide an opening message. Dr. Sydney Yee is actually our CEO of the DXD Hub. This will be followed by Dr. Emma and Dr. Sergio from FINE. They will share on the latest developments in ACTA and diagnostics testing. And lastly, we will then invite our guest speaker for the day, Prof. Wang Linfa, to share on the importance of serology testing in the management of the next phase of the COVID pandemic. Without further delay, let me introduce today's first speaker, Dr. Sydney Yee. Sydney is our founding CEO, and under her leadership, DXD Hub has helped develop a series of diagnostic tests, including several COVID diagnostic products which were deployed in Singapore's pandemic management in the past 1 year. Sydney is a serial entrepreneur, and prior to joining DXD Hub, she had co-founded and helped to grow several startups in the biotech and medtech space. As a passionate innovator of novel technologies, she is committed to helping entrepreneurs and enterprises navigate the business challenges faced in the medtech industry. Sydney, over to you.
Thank you very much for the introduction, Yuan Lu. Welcome to the webinar today, which is jointly hosted by DXD Hub and FINE. Today's session is also supported by our partners, ASEAN DX Initiative, Duke-NUS, and A-Star Central. Can I have the next slide, please? So a little bit about DXD Hub and ASEAN DX Initiative. ASEAN DX Initiative is co-chaired by DXD Hub and ASEAN NDI of the Philippines. This initiative focuses on delivering diagnostic solutions to ASEAN member states by working with innovators and innovations from ASEAN countries and beyond. We're very happy to have such enthusiastic response to participate in the webinar today. Unfortunately, after 1 year, COVID-19 pandemic remains a critical health and socioeconomic concern. So we need to have continuing to have a knowledge-based management of this pandemic. We hope the discussion today will catalyze more activities and interactions that will ultimately lead to more diagnostic solutions. DXD Hub is Singapore's national platform for diagnostic solutions. We focus on speeding up the translation of intellectual properties from innovators to users. So the speed to market is the key that we focus on. Therefore, our expertise lies in the diagnostics design and development, the production and market deployment. Obviously, to help manage the pandemic, we had in the past 12 months worked with multiple partners. We couldn't have done it just by ourselves. Working with multiple partners, we've been able to take several COVID-19 products to market, from molecular to serology, from lab-based to point-of-care tests. When it comes to market deployment, market access is a barrier that encompasses many aspects in the supply and in the value chain. So we're very grateful today to have our partners FINE to talk about their work in developing market access of diagnostic solutions, and specifically their experience in rolling out antigen rapid diagnostics tests through the platform Actor. And management of COVID-19 pandemic is about testing, tracing, treating, and preventing. So with the rollout of vaccines globally today, we are very excited and very honored to have Prof. Wang Linfa of Duke-NUS to take us through how the impact and implications of vaccine can be optimized. So on that note, I'll hand the session back to Yuan Lu to introduce the next speaker. Thank you.
Thank you, Sydney, for the introductory message. Next up, I would like to introduce our speakers from FINE. Firstly, Dr. Emma Hannay. Dr. Emma is the Secretariat of the Diagnostics pillar of the Action for COVID Tools Accelerator, which is otherwise known as ACTA, where she leads the coordination and facilitation of the group. Dr. Emma is a Harvard-trained public health doctor with a focus on global health strategy and delivery. Her work has been centered on organization strategies at a global level and implementing health systems reforms at a country level. Prior to her role at FINE, Dr. Emma had led teams at Accelerator and Unit8, which she has helped to improve the primary care for more than 150 million people across the South Asia region. Also, we have Dr. Sergio Carmona. Dr. Sergio is the Chief Medical Officer for FINE. He is an experienced pathologist with extensive knowledge in molecular diagnostics and treatment monitoring for early infant diagnosis, HIV drug resistance, and associated co-infections. His recent work involves using algorithms to generate patient cohort data sets used to monitor HIV treatment programs in South Africa. He is responsible for the evaluation of several high-throughput molecular technologies, point-of-care solutions, and assays for the National Health Laboratory Services in South Africa, which is done in partnership with the US CDC and WHO. Dr. Emma and Sergio. Dr. Sergio, over to you.
Wonderful. Many thanks, Wang Lu. What we're going to do is I'm just going to give you a brief couple of minutes background on the Accelerator diagnostics pillar, and Sergio is going to take a little bit more time talking through some of the latest advances in diagnostics that we are focusing on through ACTA and as FIND. And I think that might be a sort of particularly interesting section looking forward through 2021. What are going to be some of the key areas for diagnostics that we need to engage on? So if we jump onto the next slide. So just briefly, ACTA is a partnership, a collaboration that was announced by Dr. Tedros, at the end of April last year to accelerate access to the tools needed to support the COVID response. There are 4 pillars within ACTA that include diagnostics, therapeutics, vaccines, and then a cross-cutting health systems connector. And this collaboration brings together a range of different organizations who engage in global health from both the R&D end right through to sort of supporting countries. So the work of organizations like Gavi and the Global Fund who play key roles here. If we jump onto the next slide, so the ACT Accelerator itself, as I said, has these 4 different pillars to it that are co-convened by different organizations who have different areas of expertise around those particular products. So some of them that you'll be very familiar with, the vaccine partnership, which is also COVAX, which many of you will be familiar with, the therapeutics partnership co-led by the Wellcome Trust and by Unitaid, and then the diagnostics pillar, which we'll touch on today, co-led by FIND and by the Global Fund, as well as the cross-cutting health system connector led by the World Bank and the Global Fund. So the diagnostics partnership itself works in an end-to-end way. So both from spurring the innovation, the R&D needed to get the right tools out, through to help shaping the markets to ensure access globally, in particular for low middle income countries, through to work with countries to be able to support uptake and use of some of these new tools. So I'm probably not going to dive too much in on the structural pieces, because I think what's probably more interesting for you are what are some of the goals and targets for ACTA, which we're going to cover over the next couple of slides. So on the next slide, you can see that end-to-end structure that I talked about. So from the R&D side, there's both getting the right diagnostic tests out, but also the digital tools to help support those tests. And as many of us know, be able to collect that information up and use it effectively in the COVID response. There's a market readiness function within ACTA to help shape those markets towards competitive markets where we are aiming for sort of high quality and low cost products, which can be used to help drive access, particularly for countries who find financing diagnostics much more difficult. The third area is around supply and procurement of these diagnostic tests. And in that, a group called the Supply Consortium housed within WHO is part of ACTA. And this is a collaboration between a lot of large global procurers. So groups like UNICEF, the Global Fund, the World Bank and others to be able to act in a concerted way as procurers to make sure that there is equitable distribution of tests and coordinated distribution of tests to countries in particular. And then last on the country side, to be able to work with countries to be able to build that country capacity and preparedness for implementation of some of the new products that come out of ACTA. Just on the next slide, which is my last one before I hand to Sergio, just to give you a feeling concretely for what some of the goals and targets of ACTA are. They include, first and foremost, which was really the priority during 2020, which was to get high performance, low cost antigen rapid diagnostic tests developed and available on the market. And that obviously supported by the kind of digital tools that you need to be able to deliver that. But new goals that have arisen, I think, as the science have progressed, include making sure that self-testing is available, which obviously is an advance on some of those rapid diagnostic tests, which both opens up new areas for communities to be able to control the pandemic themselves, but obviously has a lot of complexities in being a much more consumer-facing product, as well as some of the more molecular and multi-pathogen interventions, which are, I think, currently very much a focus of ACTA in response to some of the new variants that are arising. So I realized, Sergio, that actually I have a couple more slides. I'm sorry, I was going to hand to you, but I hope it's okay if I just jump on perhaps the next couple and then hand over. So just to give you all a sense of what some of the achievements are that ACTA has achieved over the last year, and just to sort of set a tone for, I think, what we expect over the next year lifespan for the ACT Accelerator pillar. So the first one, which I'm going to dive into on my next slide, is around ensuring that there is access to the rapid diagnostic tests. So being able to launch this package of support that was needed to be able to scale up these new tools. And that included the global guidance that was needed from WHO in order to help guide country policy use. It included getting better transparent information on test performance from evaluations done by FIND and others, and then the regulatory processes that help support that both support a better quality but also clearer procurement for large procurers. Perhaps the biggest visible part of this, though, was the volume guarantee for 120 million tests for the first two products which received WHO emergency use listing. And so for these two products, to make sure that volume was available for LMICs to procure, because we knew during the first phase, particularly the last few months of 2020 and the next few months, that demand was going to significantly exceed supply for these products. And if we didn't intervene, there wouldn't be supply available for LMICs to procure. And that was then coupled with an initial pot of $50 million for procurement via the Global Fund C19 mechanism, and ongoing work within the pillar to try to fundraise more resources to be able to continue to procure these tests for LMICs who perhaps won't be able to procure them directly in the market. So that gets us to, you know, the first set of products that are available, but we have been also looking forward to what products come next. And over the last week or so, there's been some announcements that have been focused on the R&D for the next generation of rapid diagnostic tests. So tests that are perhaps manufactured at scale at lower costs. This is the most recent announcement, that have more ease of use, so have a nasal indication as opposed to needing a nasopharyngeal swab, which I'm pretty sure now everyone on this call knows is, from a personal experience, is an unpleasant direct experience to have. And then also to expand the geography of manufacturing to make sure that we have a resilient supply chain for the manufacturing of these tests. So that's just some details on the package. I'm going to hand now to Sergio to talk a little bit more on what's happened in the diagnostic space and what will happen going forward.
Great. Thanks, Emma. So, could we go one slide back? Sorry, I just want to, the previous slide. Great. So, no, next one. So I want to talk a little bit about what had been achieved, as Emma said, in terms of performance, and this is with a global lens, right? So, in just over a year, we only had the official, sequencing of the virus. In 6 months later or 7 months later, we already had a rapid diagnostic tool that could detect the antigen RDT. And as it was shown in the previous slide, 2 of these products, the SD biosensor, which is manufactured in South Korea, and Abbott, also, funny enough, manufactured in South Korea, but with a different commercial network of distribution, had already been WHO emergency use listing, which is the equivalent of the FDA emergency use authorization for many other countries that are dependent on UN agency procurement. In September, WHO very swiftly also issued guidance on how to use rapid antigen RDTs and some thresholds of performance, as you can see here. And it was no small feat to accomplish these thresholds in such short time, and with an ability to provide a result, a diagnostic result of SARS-CoV-2, almost equivalent to nucleic acid testing in about 15 to 30 minutes. These antigen RDTs are very useful because not only they provide a result very quickly, but the evidence starting to emerge that they also much more, have a much stronger correlation with infectiousness. Next slide. Following symptoms onset. As Emma already pointed out, this policy, there's been advances in prices. We are seeing an increased capacity to produce rapid tests at scale in many countries. But what's interesting about diagnostics is that it's made it into the politics of the White House, into the politics of many countries in your area, but also we're starting to use diagnostics or testing tools way outside the comfort zone of health systems. Because as we know, COVID has had major impacts to the economy, to the way we live, and the way we travel, and how we even are able to go to school or workplaces. So the tools that we're trying to develop need to cater for a less traditional healthcare-based system, and they need to be robust and easy to use, even at home. So next slide. So that's 2020, which feels like the 80s right now in COVID times. But look, we want to focus now. What are we going to be working on in 2021? And we want to see if this also resonates with the work that you're doing in the ASEAN region. So next slide. So as I alluded to already, testing is moving outside health systems, and we need to be responsive to what tools could be used by, if you want, non-traditional healthcare workers. We also are very interested in making more accessible self-testing for COVID. This is not new in the field of HIV or even hepatitis C, But the difference here is the scale at which self-testing will be required, the areas that and the sampling, as Emma pointed out. Self-testing is all very good, but to do a nasopharyngeal test, you need to be pretty brave to go and scratch really the back of your throat. So, and it's not been that easy, for example, to make sure that the product is, performs well with saliva, oral fluid. So there's a lot of nuances there that have been, that the technology is advancing quite quickly. Also very important is multi-pathogen platforms that are able to detect a host of respiratory pathogens. And some of those could be done in a rapid format, or we may need to go back to more traditional nucleic acid or sequencing-based technologies. And what we're really excited is about how to use, for example, gene editing enzymes like CRISPR for isothermal or very easily accessible tools for molecular testing and identification of SARS-CoV-2 and other pathogens. So that's also coming, and we see 1 or 2 products coming to market soon. Next slide. This is just to give you an extent of the enormous pipeline of products that have been developed since the onset of the epidemic. The many products that have been developed in the US, in Europe, but the majority is coming from Asia. And those had received some stringent regulatory authority approval, as well as does WHO emergency use listing. But going back to 2021 and what we want to achieve this year is to really bring testing to settings outside traditional health posts. And we see the opportunity of self-testing being one of those that either use an antigen or a nucleic acid-based type of system. And the various manufacturers that are coming to market and find together with many partners are starting to develop protocols and systems to evaluate this. This is a little bit more tricky, so you need to be a bit more patient, because not only about analytical performance, but also how individuals with different levels of understanding or education or language are able to interpret the instructions and use this test in a safe manner, and as well as linking that data to health systems that can address the epidemic in a better way. So next slide. Also, the supply chain is very different. How can we make accessible this through pharmacies? Last night I saw that there's also vending machines that are being potentially used, etc. So going back to novel testing strategies, we need to ensure that these are designed, that are robust, that are able to be produced at low cost, and have a format that enables self-testing. They need to be produced at mass, right? Because the market, as we saw earlier last year, can easily collapse if the demand exceeds the supply, and the regaining the stop-starts of supply for diagnostic can cause major harm to countries and health systems. And they have to be accessible. And very important is the market shaping tools that can bring this cost down, advanced purchase commitments, etc. Next slide. Now it's very important and concerning many and stopping other economies already, the emergence of new variants. Variants were not unexpected, but the breadth of how many of these are actually of clinical concern is becoming much more clear. Initially, we thought that this was just only about a higher viral load, but now evidence from the UK also showing that these variants could be, actually detrimental, and mortality and morbidity may be higher than we expected, with the association of certain mutations to key parts of the genome. Fortunately, the diagnostic tools that have been developed so far are not massively affected by these variants, but we need to keep an eye on that, and hence the need for ensuring that health system surveillance are strong and coupled very strongly with ability to sequence. And that capacity in many countries is a key priority of ACTA, to first of all map it out and then to see what tools would be available to support the network that's already been doing great jobs for influenza and other diseases, but there's still much work to be done as well. So genetic genome sequencing, I know in many countries in Asia, it's daily routine, but many other countries are way behind. So again, enabling a market and tools that are accessible, ease of use, workflows that can be scaled, etc., requires still some work. Next slide. Next slide, please. And this is just, and again, these slides will be shared, but we've been carefully mapping where these variants are in the genome and how they correlate already with the diagnostic tools that are into market. So looking forward, and I don't want to take too much more of your time, and we can address this in the Q&A. There's two main areas that the ACTA will be focusing on in 2021, strengthening surveillance systems and the R&D required in diagnostics to ensure that these products remain relevant with the new changes that we see into the virus, and make sure that also these are accessible to the countries that we are focusing our efforts to. Thank you very much. I'm running out of time.
Okay, so thank you, Dr. Sergio. We will be inserting a poll right now to the audience. I think that the audience can see the poll on your screen, and please key in your answers and submit your poll. And at the Q&A, perhaps we can actually invite Dr. Sergio to provide some comments on the poll results.
Great. So we wanted, while you're voting, we wanted to get a sense of where you're coming from. Also, in your country, we would like to get a sense of what is the currently most commonly used tool to diagnose SARS-CoV-2 or COVID-19. So have a look at the questions there. And in terms of, as I pointed out, self-testing, would this be something that resonates with the countries that you come from? And we'd like to learn, from you as well, if there are priorities that we may have been missing through ACTA. ACTA has a global reach. We're working very closely with Africa, with Latin America, and many of the Asian countries too. We know that many countries are addressing the pandemic in different ways because it may be affected in different ways, and the way it keeps on, having its high and lows. So there are different interventions that may be opportune. So I don't know how much time they have to vote for this, but we're eager to see what your responses are. We also want to know what support can ACTA provide, for example, for some of your countries. And here's a little bit of a menu. I see Emma very surprised. I hope that we're on target.
Should we all end the polling now and publish the results?
I don't think so. I want to know how many have been heard about ACTA before this webinar. Maybe you need.
Yep.
Great. Wow, nice to know that people all the way from Cambodia, Myanmar, Malaysia as well represent. So some don't know what is the most common test used in the, oh, I lost the poll. Can I have a look at it again? Was that it? Okay, great. So Sydney, this is interesting as well to understand why is testing still something that, so for policy dissemination and all those groups that do a lot of, I would say, advocacy, and limited. Great. In terms of the interest in self-testing, maybe that's great to know. Some may totally please make if it's available. Great. So I think for many countries, it looks like, at least in your region, self-testing could be something to consider. I know it's in many other countries is not the case. So because they're very traditionally been concentrating in nucleic acid testing. And what support can ACTA provide? Emma, this is for you and Amy. So do you want to comment on that?
I think it's a great one, Sergio. I mean, one that we've been really increasingly aware of is how challenging it is for countries to design a coherent diagnostic strategy as new tools become available, as is often sort of immediate pressures, and also as testing moves into new settings like schools and workplaces, which countries aren't used to formulating policies on. And so I think this is an area we had identified as being a particular challenge, and it's great to see that echoed in some of the feedback here as well. And I think as well for the Asia region, the issue of price is particularly important because countries are bearing the costs of this, and there's a very much a limit on budget. And so I think that market shaping work to be able to, in a very rapid period of time, to create an effective and efficient market for these products is particularly important.
Yeah. Okay, over back to you. And we're looking forward to the next speaker. Thanks, everyone.
Thank you, Dr. Sergio and Dr. Emma. Thank you for sharing the updates on the ACTA tools and the latest guidelines on the antigen RDT, and of course, the interesting polling exercise. All right, so next up, right, I'm excited to introduce our anchor speaker for the day, Prof. Wang Linfa, to give a talk on the importance and challenges in determining the neutralizing antibodies for SARS-CoV-2. Prof. Wang is a professor at the Duke-NUS Medical School under the program in emerging infectious diseases. He is an expert in the field of zoonotic diseases, bat immunology, and pathogen discovery. Hence, he is widely and affectionately known as the Batman in our community. He played a leading role in identifying bats as the natural host of the SARS virus, and recently he led his team to develop an antibodies-based serological test for COVID-19, which we'll hear about later on. His team is currently working together with local and international partners to develop new vaccines and therapeutics for COVID-19. In addition to his research contributions, he is also a member of multiple WHO committees tackling the pandemic. Let us welcome Prof. Wang Linfa. Prof, I think your mic is muted.
Okay. All right, thank you.
We can hear you.
Yeah, thank you, Yuan, for the introduction, and thank you, Cindy and the FINE for organizing this very timely webinar. Yeah, so I mean, my take will be slightly different. I'm a basic research scientist, but I have a strong interest in really, tracing the disease and how to do the prevention. So right now with COVID-19, I can think of really 3 hot topics: vaccine, variants, and origin. I mean, today, the WHO mission in China, in Wuhan, first, after 2 weeks quarantine, they just got out of the hotel and tried to do the real sort of underground work. So I will go through all these 3 topics in the context of the importance of neutralizing antibodies. Sergio was saying, I mean, 2020 is like ages ago, and so I thought it would be good to go through that, how it started, right? So anniversary, when is the first anniversary of COVID-19? It's very hard to define because we, I think, lost opportunity to map the patient 0. So WHO 31st December really alert on this novel virus. And then in the last 1 month in January, there's lots of events. And then it ends on January 30th with the World Health Organization declare this as a public health emergency of international concern. So I had the privilege of sitting on that committee, which I cannot discuss openly, but it's a very intense. So to me, the anniversary of COVID-19 is actually tomorrow, because that's where the fake was caught. Okay, so about midway in July, Nature published this review, basically says 6 months of coronavirus, the mystery scientists are still racing to solve. And these are the sort of 5 questions that at that time remaining. Unfortunately, I think another 6 months later, most of these questions are still not very clear. So today I'll be focused on the last 2, how well a vaccine work and the origin of virus. Interestingly, because these are the 2 key questions, basically, how did it start? How could we get out? The exit strategy. And to me, I work in both of these areas. I'm biased, we just listen to find, the diagnostics, of course, the molecular test is very important in the disease control and prevention, contact tracing. But for me, these 2 areas now, I think a highly specific and versatile serology approach is required. And that's what I have been working on this for the last 12 months. The reason is that, for human now, COVID-19, human applications in terms of serology, you heard a lot of herd immunity, immunity passport, longevity of protect immunity, whether that's from infection or vaccine. Now we have vaccine, convalescent plasma therapy. And then in terms of searching for the natural reservoir, for COVID-19, we have another burden or another worry is what we call a reverse zoonotic transmission. We already know the virus can be passed from human to animals. Mink, of course, is a high profile. Just 2 weeks ago, gorillas in the San Diego Zoo, So this is actually quite scary because that can establish a new reservoir. So to do all of these, what we need is for the human application highlight in red, we need a neutralizing antibody, very specific but easy to conduct. And for animals, it's even more demanding. We not only need a specific, hopefully neutralizing antibody test, but it has to be species independent because we don't know which animals could be the intermediate host and could be the what we call the spear back host. So you have seen the image on the top left many times on TVs and on media. So this is the imaging of the coronavirus, and the key protein is the spike protein on surface. And so that is, if you look at the genome, this is the coronavirus genome. It's around 30,000 nucleotides. So it makes lots of proteins and many of regions to worry about. When you get an infection, or if you get immunization with a vaccination, it creates lots of antibody, hundreds, if not thousands. But only a very small proportion is functional, we call neutralizing antibody. Majority of them, we call them binding antibody. They just bind to different parts of your protein. Fortunately, for SARS-CoV-2, because we had 17 years of research, so because it's such a close cousin with SARS-CoV-1, so as soon as the sequence was published, we can make an intelligent guess to say the spike protein is very similar, and hence the key part, what we call is the receptor binding domain, is also mapped out very quickly. So most of the neutralizing antibodies, around 90% or more, is directed just to that particular region, the receptor binding domain, RBD. Okay, so the gold standard, if you want really specific measuring of neutralizing antibodies, you have to use what we call virus neutralization test, VNT. And for this, you need a live virus. So the live virus comes in, binds to the receptor, and if it's successful infection, you call CPE, cell death. In the presence of neutralizing antibodies, hopefully it's potent enough to block that process and the cell is healthy. So up until today, this is still the gold standard, but it's only gold standard for specificity. In terms of sensitivity, the safety, the operation, the cost, it's not a gold standard, far from it, actually it's at the bottom. So it has one advantage and lots of disadvantages. So to address both to get a neutralization antibody, but also have a species independent manner, we invented this surrogate virus neutralization test. So again, Sergio said, it's like ages ago. I just was thinking, counting months, our paper went out in April. So that was 9 months ago. And so this was published in Nature Biotech. The core part of the invention is what we were doing is try to do a biochemical simulation of this very cumbersome and costly virus neutralization test. So this is the diagram as I have shown you. This is the SARS-CoV-2. It binds to the ACE2. If it's successful, go into the cell, that's the cell surface, and cause CPE. In the presence of functional neutralizing antibody, you block that, and we say it's positive. You have virus, you have the neutralizing antibodies. So what we did is using protein engineering. So first, the receptor is a membrane protein. So we express only the soluble functional domain. So we can purify easily and coat on a very, very common ELISA plate reader, a plate. So for the virus, we did not even use the whole spike. We just clone and express the RBD, the receptor binding domain, the RBD. To make life even easier, we chemically conjugate the host reciproxase. That's the enzyme, you know, the audience, anybody in the audience, you have done ELISA, you would have used HRP. So now this protein will bind to the AC to already immobilize on ELISA plate, and then when you add a substrate, you get a color. If you have a neutralizing antibody, which is on the right, and then just like the virus neutralization, you don't get that binding, and then the signal is weakened or is eliminated. So it's basically inhibition assay. Papers are published, I just highlight how good it is. So this is a correlation. This is a live virus we call conventional virus neutralization titers in logs. So this is a 10, 100,000. And this is a surrogate virus neutralization, same scale. As you can see, the correlation at about titer 10 above is very good. But when you get to the bottom, when you get really low, of course, there is some discrepancy, and seems like the biochemical assay is slightly more sensitive. But the R square is 0.86. That's pretty good, consider that one is a biochemical, one is a live virus. And the specificity reaches 100%. We use two different cohorts, one in Singapore, one in China. And the sensitivity, these are the sensitivity really marked against PCR positive. So it's 98%, and that's pretty good, as we know that some of the patients that PCR positive never develop neutralization antibodies. I will show you the data later. In terms of species independent performance, in the Nature Biotech paper, we use rabbit sera and the mouse sera. As you can see, the negative does not inhibit, so this percentage inhibition, and this is a titration. And it goes from high inhibition in a dose-dependent manner, and it's a beautiful curve. Since then, we have passed our test to more than 30 different countries, and our collaborators all over the world has used this for human mouse, of course, but you have pangolins, bat, minks, hamsters, raccoon dogs, pigs. 14 animal species have been used and all perform really well. So that's another beauty of the test, is that you don't need any modification. You can go from human to bats to rabbits all the way. And with the help for GeneScript is our commercial partner. They were very quick to manufacture this kit. And then the DXD A-Star, under the leadership of Sydney and her team, helped with the validation approval. So this kit, commercial kit, was launched on May 15th, 2020 in Singapore. And this is a very easy test. You can be done. Any lab you have an ELISA reader, and so whether it's research or clinical labs, and it's only in 1 to 2 hours. So I had to go myself. So if I can do it, I think anybody can do it. And on November 6th, we got FDA approval. That's a huge milestone, because in the history of FDA, they really never approved neutralization tests, because the classic neutralization test requires use of live virus and the live cell. So it's very hard for FDA to approve that, because the quality assurance is difficult. For COVID-19, for those of you working in the field, you know that there is another surrogate called a pseudovirus-based neutralization test. So pseudovirus basically use VSV or HIV backbone, and you pseudotype with the spike protein. But even for pseudovirus, you still need to use a live virus and a cell. So FDA also has not granted any EUA approval. So it's the first and remain the only test which can determine neutralizing antibody. So again, referring to Sergio's comment, the speed, for COVID-19, this is COVID-19 speed. You really had to be very, very fast. So March 10th, I discussed my invention with our commercial department, and 10 days later, we get a proof of concept. 25th, patent filed. 26th, negotiation with GeneScript started. And less than a month, we got a beta testing out and approved, launched, and FDA approved. So from a concept to a commercial product was in 70 days. So now I'm going to switch to really to ask the questions of, can antibody test protect immunity? Because for the COVID-19 exit strategy, I think to define a protect immunity biomarker is almost essential. So in order to address these questions, actually, you have to address two separate questions. The first is in the other EUA or CE marking approved tests, ELISA, LFAs, rapid test, all of these are binding assays. They measure binding antibodies. So the number one question is, can binding assays like ELISA accurately measure true neutralizing antibodies? The second question is that then if you can quantify neutralizing antibodies, can these be used to correlate protection? So two different questions, and I'll address them separately. The first is that, as I said already, RBD is the main target for neutralizing antibodies. So it's reasonable we focus on RBD. So that's what we did. So to illustrate this point even more conclusively, I would show the data with monoclonal antibody, although we have data polyclonal, but I don't have time to show. So this is a monoclonal antibody from mouse, 4 of them, selected with decent binding with RBD. Okay, so this is RBD. So that you concentration dependent, and that's the OD. As you can see, all 4 of them binds well. The red one is the best. Now you come to our CPAS or the surrogate virus neutralization. So the same RBD protein, the protein is the same. The only difference is now we're looking for the ability of this antibody to block its binding to ACE2. That's the surrogate neutralization. Now only 2 can do it, and the other 2 fail. The best binder, the red map, now is negative. Same results for human monoclonal antibody, 4 of them. As you can see, the yellow or orange one is second in binding, but total negative in blocking, in neutralization. The other thing we also noticed is that if you mix these 2 monoclonal, the green and the blue, and then if you mix equal molar concentration, in theory, the predicted curve should be in between, right? The blue is here, the green is here. But what in real data you got is the purple. That's what we call is neutralization epitope synergy. So when you have 2 different map bound to 2 different neutralizing epitopes, if they're both in presence, 1 + 1 is greater than 2. So this is what we have been observing, because with our CPAS neutralization test, it gets much more sensitive because the test is independent of epitope, independent of a species, whether it's animal or human, independent of isotype, whether it's IgG, IgA, IgM. As long as you have neutralizing epitope, they will neutralize and they have a synergetic effect. Okay, so the second question is, does neutralizing antibody really predict or protect immunity? And we said yes. Okay, so this is from past experience, but even Fauci, he was directed this Moderna vaccine trial in phase 1. So that was in June, on June 1st, that's more than 7 months ago. And he got really excited. The thing that made everybody be cautiously optimistic is that we did not just see binding antibody. It was clearly that antibody that was neutralizing the live virus. So he thinks that's a reasonable assumption that it's going to be a correlate of immunity, protect immunity. I mean, that was in June, it's COVID speed, right? So we have many, many papers now come out, really demonstrated correlation between neutralizing antibody and protection. I still think the earliest study is this one. It's the most beautiful, although it's a very small study. So this is a study carried out by the Washington University in Seattle. There is a fishing sort of exercise, a fishing vessel went out for sea, and they had 122 people on board, and they were able to sample that 120 of them, and they did a PCR. It's all negative by default. If it's PCR positive, they will cancel the trip, right? And they also did serology, really credit to them, So they did serology, and here's the data. Out of 122, mostly are negative, as you would expect. I used the Abbott test. I don't want to, just put Abbott there because this is just published in the paper. So the Abbott IgG binding assay give you 6 out of 122 positive in antibody. And using the ACE binding blocking, that's the CPAS from GeneScript, only 3 are positive. And the inhibition is vary from 84 to 93. The other 3 is negative. When they come back, 85% of the crew members were positive by PCR. So the attack rate is high, 85%. They're in a closed environment on the same ship, that's why. But very importantly, although it's 85 attack rate, out of 6, 3 were positive and the CT values are low. So it's very high positive, but the 3 that had a neutralizing antibody by our CPAS is negative. So that's, as I always say, it's a beautiful study, although the sample size is small, but it's a beautiful, almost like human challenge study by stealth. last months, now maybe just a few weeks ago, there is a Nature paper from a Harvard group. So this now is an experimental sort of study, and of course you cannot do it in humans, so that's done in non-human primates. You immunize the monkey, you get neutralizing antibody, and you then passively transfer this to naive monkey, then you challenge with live virus, okay? So conclusion is that neutralizing antibody alone is sufficient to provide protection and in a dose-dependent fashion. When you do the titration out, they found that actually relatively low antibody titers are sufficient for protection. Hence, they concluded that cellular immunity, T-cell immunity response may contribute to protection when the antibody response is really low and suboptimal. And this is a New England Journal of Medicine publication, so led by Oxford. So this is involved like over 12,000 healthcare workers. They just did a longitudinally sample them, bleed them and test serology. And this one, they did not use CPAS, just using binding antibodies. But the conclusion is that for those antibody positives, they don't have a symptomatic infection, okay? So it's clear that, antibody, especially neutralizing antibody, is protective. The other hot topic in the media and for scientific community is immunity longevity. I mean, if you have been following the literature, in June to August, people will panic because there are papers with headlines to say neutralizing antibody only lasts for 3 months. And then in the last week or so, then you have a headline say, don't worry, the neutralizing antibody or protect immunity lasts at least 8 months. I don't want to criticize this, headlines, but I think they only report really a very sort of biased headline because the real scenario, both are right, but both are wrong, okay, in that. So what we did is a longitudinal study in Singapore, a cohort with all the COVID-19 sort of recovered patients. 164 of them were really nice enough for us to follow them for 9 months. And that's pretty longitudinal. And what we discover is the neutralizing antibody waning sort of dynamics is multi-faceted. It's not like 3 months or 8 months. And it actually lasts from days to decades, okay? So you will ask, how do you know it's decades? I will show you the data. We have AI machine learning modeling. So this is the data using CPAS to measure neutralizing antibody only. Even nowadays, I don't care about binding antibodies because that's the functional antibody. Days post symptom onset. One group, negative, never had a neutralizing antibody. Another group, yes, positive, goes to all the way to 90%, but then they wane very rapidly. We call rapid waning, slow waning. And this one is persistent in the sense that most of them, their antibody really does not change much. The last group we still don't understand is a delayed kind of response. Their antibody is still going up, 6 months later, okay? The two sort of extreme group, the negative and delayed onset is minority. Plus, I think that's not going to really tell us about the longevity. So we focus on the 3 groups in the middle. Using the data from the first 6 months, we developed an AI machine learning prediction model. And we submitted a paper. And then during rebuttal 3 months later, we had the data from the 9 months. This gave us a chance actually to validate our prediction model. And that was pretty good because the concordance of the R-square was close to 0.9. So 90% accuracy, that's pretty good. So what you can see is in this group, now we're talking about neutralizing antibody longevity. So from positive to negative, days post-symptom onset. The range goes from 35 to 150, 48 days, and the median is 96. The next group, median is 200. The next group is around 600. But I'd like to draw your attention, the extremes. So we have patients with neutralizing antibody positive, 35 days later, neutralizing antibody negative. Using our AI machine learning prediction model, the top of the range is that at least a couple of patients recover. They will have their neutralizing antibody last more than 14,000, close to 15,000 days. And that's 41 years. So 35 days to 41 years. That's what I try to say. If you see in the media to say that immunity lasts 3 months, not right. Immunity lasts 8 months, not right. You have to know yourself if you get vaccinated or infected because immunity varies so much, depends on the individual. So the origin of SARS coronavirus, I'd like to make a statement because it's so much politics involved, and I want to declare that we should know this for science, not for politics. So that, the WHO mission started 2 weeks ago to China and finally, and there's a lot of report and already says, it's not about finding China guilty. I think, this kind of a title is actually drawing more attention to that. And that, we have local media, so say, don't expect a conclusion from WHO's China visit, and says the expert, it's a very famous expert in Singapore, and Professor Dale Fisher. So I think I agree with Dale is that, don't set an expectation too high, because I was personally involved 17 years ago in the same mission. So 17 years ago, as you know, SARS-CoV-1 started in November 2002. So, but the international community worked together, and plus the virus was less transmissible and more severe. So we can, contact tracing and the quarantine much easier. So the outbreak was controlled by July, but even that, we still did not know how the virus started. So WHO assembled a mission on the reservoir and early transmission events in August. And I was privileged enough to be selected as one of the eight scientists on the mission. It's so different, right? Now before the mission, so much coverage and who is on it and who is not on it, why that person should not be on it, that person should be on it. When we did that 17 years ago, there was zero report. Nobody followed us. That's give us all the space to do science. And I want to single out one participant, Ann Shuka from the middle, in the middle. Ann Shuka, is a very active epidemiologist from CDC, and she became a deputy director, and she does the briefing in White House, on regular basis. But that team 17 years ago, hospitals, even military medical institutes. So imagine, today, I think that's not gonna happen. The other message I want to really for you to take home with is that the mission is about knowledge exchange and brainstorming and discussing what's the new approach we can use. The mission does not do real science in the sense that they're not going to sample, they're not going to PCR, they're not going to do neutralization test. So from that mission, I was able to organize an international consortium from 5 nations, 3 nations, 5 institutes, and then 2 years later, we discovered the bats as the origin. So I want to emphasize, as Dale Fisher says, don't get your expectation too high because the real work will start after the mission, okay? So what's the best approach to trace the origin? Of course, the young scientists in the audience, you only know about PCR screening. Everybody does PCR screening to trace the origin. If you're rich enough, you do next generation sequencing virome, so discovery approach. And now for COVID-19, there's a lot of discussion about we should use ACE2 to guide the surveillance for origin. The reason is that ACE2 is a receptor, and now there are so many genome sequences of mammal species on Earth have been published. So there are people are, functionally interrogate ACE2 in the, 50 different animals and to see which is susceptible, and then think that that's the most likely could be the origin of the intermediate host. I just want to sound a caution about this because, virus evolve in a fashion that regardless of which receptor they use. Okay, so I just want to give you this phylogeny tree. This is the S gene. Based on the whole S gene, and we can now clearly have 2 lineages. One is the old SARS-1. So this is the Beijing 01, and this is the back SARS coronavirus. So all of these are the same species called the SARS-related coronavirus. And now we have a new lineage, is the COVID-19 as the prototype, and there are already many bat and pangolin virus. So I call this SARS coronavirus 2 related coronavirus. So they're the 1 and the 2 lineage. So for S gene, it's clearly there's 2 different lineage, right? But for the RBD usage, if you zoom to the RBD domain and then you do a phylogeny, then you get a different tree. These guys all use ACE2 as a receptor, and these ones don't use the ACE2 receptor. So now these lineages are not the identical one. So I'll just give you an example. These are the 2 different bat viruses, one from Japan, one from Yunnan province in China. So using the whole spike protein or gene, they're very close. But if you do the RBD, they're in 2 different lineages. One uses ACE2 and one don't. So with that, I think, my favorite is to use the antibody, especially neutralizing antibody should be the leading tool to trace the origin of the virus. And again, I'm not making this up because it's speaking of my experience for the last 25 years, so I was involved for the origin of Hendra, Nipah, SARS, MERS, and serology played a leading role in discover both the intermediate host and the reservoir host. So, I can predict, and I already have data, serology will play a very important role to trace the origin of SARS-CoV-2 and intermediate host and early transmission event. So you have seen many, many discussions. This one was published again in July. Two leading scientists, Peter Daszak from EcoHealth and Jeremy Fry is the head of Wellcome Trust. Both argues to say the hunt for the origin of SARS-CoV-2 should look beyond China. And the reason for that is that nobody's arguing these are ancestral virus of SARS-1 and SARS-2 come from bats. So you look at the bat, diversity and species richness, the darker the red color is, the more rich, right? So Yunnan province is here, Wuhan is here. Wuhan has bats, but it's not really well known for bat diversity. Southern China, Yunnan province, that's where the close related bat virus was discovered. And then you go across the border to Myanmar, to Laos, to Vietnam, and all the way down to Cambodia and Thailand, then even to Malaysia. You see all of these bats, very, very diverse. So we got a WHO fund. So WHO had some emergency funding end of last year. So I was a PI on one and co-PI on two, all based on our proposal to use CPAS serology to lead the work to trace, investigate the SARS-CoV-2 related virus in animals. So our project is focused on Southeast Asia and start with Thailand. And the one read by EcoHealth Parish is internationally, So we have a paper just accepted. I could not talk too much yet, is that we now have evidence of SARS-CoV-2 related coronavirus is circulating in bats and the pangolins. Yes, pangolins will be in the media again in Southeast Asia. Last topic is variants, right? Sergio already touched, it's a hot topic. So in terms of variants, there's really two questions. The first question is where the current vaccine protect against infection by VOCs. And there are so many papers already published, even BBC says, Moderna vaccines appear to work. And then there are papers with the Pfizer vaccine, using the serum from vaccinees, and they demonstrate that they can neutralize the virus. And there's another talk about, serum from the Pfizer and on this variant. And the African variant, these are the UK variants, and the African variants are just out. So I think the bottom line is that, yes, these are variants, and there are some impact, but the impact is negligible. So both companies says we're not going to change the vaccine because the difference may be only 3-fold and 6-fold difference. And the natural variants, I show you that the neutralizing antibody variation between infected, among infected patients vary a lot. Now we also have this data on the vaccinees. The neutralizing antibodies varies 20 to 30-fold, So the individual variation is 20 to 30, and the variance, give you the variation maybe is around 3 to 6. So relatively speaking, it's not a big issue. Okay, the second question, which is also very important, for the field and for myself, is where neutralizing antibody tests like CPAS be affected. So can CPAS still detect neutralizing antibodies from infection by VOCs? So I thought I should just do a little bit of science behind that. This is the RBD, receptor binding domain sequence, and the evolution. So this is the first virus, the Wuhan HU1. This is the UK variant, this is South African variant. This is the bat, closest variant from bat, and that's a distant variant from bat. As you can see, compared with the bat RBDs, these guys are basically identical, right? And it is true because this is, if you define the Wuhan one is 100%, UK is 99, and the South African is 98, and then the closer bat is 91, the distant one is 66. In terms of ACE2 usage receptor, the Wuhan one uses good, but the variant even better binding. The bat one of them is weak binding, one of them is negative. Okay, so here the sequence, and you can see the top one is the Wuhan, second is UK, and the third one is South African. So basically, there's only two sites. The 501 is here, and then the 484 is here, okay? So the back variant, as you can see, many difference here. Okay, so in order to really appreciate that, what the impact will be, ideally, I always say we need a 4-way cross surrogate virus neutralization test. So we need a wild type sera. We have a lot, in Singapore, we have, 200 patients, so we can do that. And then we have the CPAS use the RBD from wild type, and now we have a CPAS use the variant from UK. Ideally, what we need is also sera from patients from VOC. We don't have it, the blue arrow, so I cannot present the data. So the red one, we have done it. So this is the wild type RBD, which is controlled in green, and this is the 501, the UK variant. As you can see, the binding affinity that the RCE2 RBD binding, it's a slightly better, the variant. But when we express this and do the surrogate virus neutralization, so here is the wild type percentage inhibition, and here is the mutant. As you can see, it's almost as good, or if anything, it's slightly more sensitive. We don't understand, but the conclusion is that I think the variant is not going to impact the CPAS performance. So what we did is we go further. What we did is use these two rabbit RBD, because the bat coronavirus RBD, we don't have the sera from natural infection. So what we did, we purified the RBD and we did the rabbit immunization. So these are hyperimmune rabbit sera. As you can see, both still can neutralize the virus in the CPAS use the wild type sequence. Of course, the percentage inhibition now, two rabbits is like for this one is like a 90 to 70. This one is good to 70 to 30. So there's an impact, but I think I'm not worried about, because the variant can never be so variant to reach the RBD sequence as the bat molecule, because if we get to that, then the virus will not bind the ACE2 and it will be eradicated from the human population. So no matter what variant is emerge, they still bind to ACE2 receptor, and I think that CPAS will still work. So I try to wrap up, and in summary, so what I think what we are here now is that in terms of neutralizing antibody and protective immunity, here are the facts and the current status. There's a lot of discussion about relative importance of neutralizing antibody versus T-cell immunity. My diplomatic answer is both play a very important role in protection, and the antibody plays a more important role in prevent infection transmission, whereas the T-cell is a latecomer. By the time T-cell is activated, virus is already replicating in your body. So T-cell is more important maybe to dampen the disease. So that's like a basic science. On a practicality sort of for ground, there is no assay suited for large scale T-cell immunity test. Just like the live virus neutralization, FDA will never approve a T-cell test which use live cells from blood. And for natural neutralizing antibody test, right now we have 3 technologies. The classic virus neutralization test, as I presented, you need a live virus in the BSS3 lab. And there is a pseudovirus-based virus neutralization called a PVNT. That's used a BSA2 lab, but because it still uses live virus in the live cell, FDA, I think, will unlikely approve that. And then the third one is what we developed called surrogate neutralization, and the commercial is CPAS. It's approved by HSA first, and then EU, FDA, and others. The other thing I think of course, will play a very important role is that to standardize this, right? Normalize and standardize it. So finally, we get a WHO, really start to issue this standard sero panel, with predefined international units. For the doctors and the healthcare workers in the audience, you're very familiar with that, right? For hepatitis B vaccination, for example, just vaccination is not enough. You need to do a serology test so that you get a report of international units. For Hep B, it has been around for 30 years. That's why the IU is well established by WHO. So you need to reach that IU to be able to work in like my lab. So if I hire a postdoc to come to my lab, vaccination is not enough. They have to do a test, and the IU has to pass that 10, units 10, that's the threshold. If you're below that, you have to get a boost. So I think that's going to happen sooner or later for COVID-19. So what we're working closely with the WHO and others, is try to calibrate the international units panel from WHO, but that one has a limited, supply. So what we need is a calibration kit, and we're working with GeneScript. And so the CPAS, now we will sell 2 kits. One is a CPAS kit, and hopefully another is a CPAS calibration kit. And then you can calibrate your CPAS rating, become international. The last one now, is the holy grail of the exit strategy. Eventually, what we need is a correlation of the international units level with protection, just like for hepatitis B. The research is ongoing. I'm hopeful with mass vaccination now ongoing in different nations. Hopefully in another 3 to 6 months, we will get that. And once that is established and the IU is established, then we can really talk about exit strategy. What's that exit strategy? Immunity passport, right? A COVID-19 pass. So it's still controversial. I mean, WHO is not going to come on board with that until more data is available. But for me, that's the only way out, because to get a herd immunity of a 70% protection, first, you need a biomarker, right? Correlate. How do you know 70% of the population is protected? Because 70% of the population is vaccinated does not mean protection, okay? And then the most sensitive industry of course is travel, right? So, Qantas, Australian, the carrier, basically even in November, way before any mass vaccination, went to the government and they tried to say that, all international travelers to board Qantas, they need to see a piece of certificate say you're vaccinated. Personally, I think, I agree with this only for 2 reasons. One, vaccine does not offer protection, not 100%. Secondly, what happens if I'm infected and I have high neutralizing antibody? I don't want to be vaccinated. So I don't have a piece of vaccination certificate. Are you going to block me from boarding Qantas? Okay. So my dream is for CPAS to be a verb. Currently the verb is, are you vaccinated? Okay. To me, I think we can go even higher, is that you have a certificate, are you CPAS? Once you're CPAS, we don't care whether you're vaccinated or you recovered from infection. So with that, I'd like to thank you for your attention. And I have to declare the conflict of interest because I'm a key inventor of the patent, and the kit now is the market under the trade name of CPAS. Thank you for your attention. And my team, of course, very happy and a hard work team. Thank you.
Thank you, Prof. Wang, for the very, very interesting talk. And we have had so many questions coming in. And wow, I think we have to start immediately. Okay, so let me just start with a question for Prof. Wang.
Mm-hmm.
Prof. Linfa, what is your opinion on using a vaccine that is perhaps 62% effective, given the wide variability in recipient response? Are those given the AZ vaccine going to have to be vaccinated again? Say with the Pfizer vaccine, will persons vaccinated will have to have the neutralizing antibody test?
Yeah, so this is a tough question. Half is political because WHO basically set the benchmark is 50% efficacy. And I can understand, I work on the committee, but I'm speaking as a personal view. So I try to declare the conflict of interest. I'm not speaking on behalf of WHO. Because that we are in a very, very dire consequence. If we don't have any vaccine to use, I mean, we really cannot exit. That's why the bar is set at 50%, because the 50% efficacy, if you, vaccinate the whole population, at least you dampen this, to a significant degree, okay? So that's why the 60% is above that 50% benchmark. That's why we say anything above 50% in theory, that WHO will, give a pass, and that become a political decision by each nation to use them. Okay. So the second part of the question is that consider variation of this, whether you take AstraZeneca, you need a boost with another vaccine. I think that's, again, is a sensitive question because even for Pfizer and Moderna is 95 to 90 protection against disease. So we don't know what's the protection against infection or transmission, So without pushing my agenda too much, my view is whether you're vaccinated with Pfizer or AstraZeneca or Sinopharm, you have to CPAS yourself first. Because if your neutralizing antibody is at a high level, no matter which vaccine you use, you don't need to boost. But even if your neutralizing antibody is very low or negative, even with the best vaccine, you may consider to boost. A little bit politics and a little bit science there.
Thank you for the response. I think related to this, and probably a question that is more personal to me, there is a question from the audience. How realistic are countries to allow for travel if people are vaccinated? Right? And what do you think it needs to be done before the governments will allow for this to happen?
Well, I'm vaccinated. I did my first injection last week. And so in 5 weeks' time, I'm vaccinated. But of course, for me, I'm going to CPAS myself, right? I'm going to not only vaccinate, but CPAS. Okay, hopefully I'll be passed. So again, it's a very difficult, question. And I can tell you that, I've been consulted by government agencies from at least 5 nations. And I said, scientifically, as soon as we have this IU for protection, I can advise you. But at the end of the day, it's a political decision each country have to make. For WHO to mandate, to say you have to have the IU before you can travel, I think that will never happen. It's too sensitive. So at the end of the day, is science plus political will of each nation, then will allow us to travel. Because right now, to be frank, there's no incentive to vaccinate, right? If you're vaccinated, if you travel, you come back, or you go to another country, you still get a quarantine, regardless of if you have vaccine or no vaccine, okay? So I think until that policy is changed, then I think the incentive for vaccination right now for countries like New Zealand and Singapore is very minimum. But for the country to open that based on vaccine or correlation of protection, then you need both science and a political will.
Can I use my panelist voice here? Because Prof, that is a really good question. And I know there are lots of questions for you on the chat, but from a diagnostic point of view, what do you think some countries already using, the 72-hour PCR only provides certain coverage, for traveling, but now some countries that are adding, for example, antigen RDT 4 hours before loading. What are your thoughts on that? I mean, it provides a bit of as you will point out, many will still be left behind without a vaccine. So for the audience and those interested in going back to normality, what do you think of that?
So this is where your sort of talk is important, because your talk was a project to the future, says it has to move out and become, self-testing, home-based testing. Until you do that logistically, because, a rapid test is good, but how frequent you do that rapid test, right? So, we have so many cases that negative before boarding, negative on arrival, positive during quarantine.
Yeah.
So unless, so the whole exit strategy is abolish quarantine. So to abolish quarantine, you have to be really sure that you're not in the incubation period, so you're negative. No matter how many times you test, whether it's RDT or PCR, it's negative, but then 7 days later during quarantine, it becomes positive. So this is where I feel like, I'm biased again. So for me, is that, for these people who are not already infected or immunized, there's almost no way out, because if you're the naive population, no matter how frequent you test, that if you're in incubation period, then there's always a risk. That's why all countries still want to have a quarantine, right? But once we have a correlation protection, then at least for a small population who recover from infection and vaccinated has a high neutralizing antibody, and if we have a scientific evidence to say, IU, 100 and above is safe, then that will be the way out.
Can I ask you last question regarding your T cell assay, right? Because some vaccines, mRNA vaccines, may not be producing strong neutralizing antibodies, but the clinical evidence is starting to show that they are protective, right? And we know that immunology is not just about B cell response and generation of NAbs. What do you think about the value of concentrating some effort on a T-cell assay that could be, easy to use, fast, a bit like the iGRAS for TB, that covers this whole other area of protection that it may not be purely based on neutralizing antibodies?
Definitely, definitely. I think the way out is that, do neutralizing antibody test that's easier, cheaper, and then you basically segregate to the high neutralizing antibody level or low or negative. For the low or negative, you have two choices. You get a boost or you get a T-cell test if the T-cell test is commercially available.
Great.
Yeah.
Emma, that's for your Q3 of 2021, a quick T-cell iGRAS-based assay. Same price.
Same price.
Okay, I think we probably have time for maybe one last question, and I'm just going to take the most recent one that's asked, right? The comment says like, it is interesting that with SARS and MERS, there was not as much cry or call for vaccines for the masses as compared to the COVID-19. I just lost the question. Where is it? Yeah, do you foresee this COVID-19 vaccine is something like a requirement as with the MMR, etc., and other vaccines currently mandated?
Yeah, it's a good question. I think, this is where, my own opinion changes so much. This time last year, I was in Wuhan. When I just came back from Wuhan, I said, the worst is we have another SARS and we can eradicate the disease. And then my view changed to say, okay, now maybe we need a vaccine to really contain it. And now I'm in the view, maybe even vaccine could not help it. And then we have to co-live with it. If we co-live with it, and then if the virulence and the transmissibility, so the variants right now, unfortunately, is going the opposite way. We are hoping there's a variants become more transmissible, less virulent, and then become a herd immunity, natural immunization. If that does not happen, if the variants is going the way like the South Africa and the UK right now, then I think that it's almost, you can envisage that like an MMR vaccine or Hep B vaccine, every child born in a high risk area, you have to be COVID-19 vaccinated, So that's obviously the worst scenario, but it's not out of question. So my kind of optimistic view ended basically.
But it could be a multivalent vaccine as well, Prof, right? Because maybe we're just targeting one and there's still room for adding to it.
Especially with mRNA vaccine, I mean, the beauty of mRNA vaccine is incredible, right? You can not only rapidly deploy, and mixing is easy, just change the sequence.
Great. I think we really have run to the end of this, this webinar. But before I end, I would like to invite our CEO, Sydney, to perhaps say a few words and close the session. Sydney, please.
Yeah, hi. Just want to thank everyone for participating in this webinar. I want to especially thank our speakers, Emma, Sergio, and Linfa. Listening to Linfa talk, it's always very satisfying because Linfa always answers so many questions, but it's also unsatisfying because there's never enough time to ask more questions that I have. And so I'm sure I see many questions that are left unanswered. Please feel free to contact the organizers for questions that you have and you would like us to follow up. There are also a lot more other questions that are for FINE and for ACTA that I see that Emma and Sergio have been sort of separately responding to. We are very happy to follow up with some of the other questions that have not been answered. And this is going to be sort of the first of a series of webinars that we will jointly host with FINE and ACTA, and we hope to be able to see you guys in our future webinars. Thank you very much for the participation, and thank you again to Sergio, to Emma, and Linfa. A fantastic session. Thank you.
Thank you.
Many thanks all, and many thanks for the great questions and discussion.
Yeah. This was excellent. Thank you very much, Sydney
Okay.
and Linfa.
All right, thank you so much for your participation. It has been a wonderful time. Thank you again to the speakers and all the participants joining us. I'd like to wish you a good day ahead and keep safe until we meet again. Thank you. Bye-bye.